Hi, everybody. So my name is Anne-Sophia. I'm from the University of Limoges in France, and I will speak today about the 3D cellular models of myelination using human-induced pluripotent stem cells to study CMT4C. CMT4C. Sorry, everything is okay. Okay, so you know that Charcot-Marie-Tooth is a disease that touch a peripheral nerve system. And there are more than 100 genes involved in this disease. And today we will focus on SH3-TC2 that involved in the demyelinating form of the disease. So it's not like PMP22 that is autosomal dominant here. It is autosomal recessive, so it means that we need to have two mutations to be affected by the disease. And the most frequent mutation is this one that is present in almost 80% of the patients. So our main objectives are to better understand the pathophysiology and to test therapies. So to do that, we need models, and I will show you previous SH3-TC2 models that exist. Most of them were from animals and especially from mice, and there were cell lines that were either neuronal, either non-neuronal, and were transfected. There were also primary cells, mostly from a mouse or rat, dorsal root ganglia and Schwann cells, and also in vivo models, a natural one. So there were dogs with mutation in SH3-TC2. And also there is a mice with a natural spontaneous mutation, and also the model that is knocked out. And for human, we've got only non-neuronal transfected cells. And there were no human cells models expressing mutated SH3-TC2. So we wanted to create them, but we couldn't remove Schwann cells directly from the patients. And so we decided to create these cells using the iPS strategy, but also the CRISPR-Cas9 strategy. So here I show you the development, and these cells are the embryonic stem cells that will give a fetus and then an adult body. And in this adult, you will have a differentiated cells. And there are a very nice strategy named iPS strategy that allow to use differentiated cells to create induced pluripotent stem cells. And then when you've got that, you can differentiate these cells into neurons or Schwann cells. So it's what we did in our lab using fibroblasts from 5 controls, and we obtained control iPS cells. But it was very time consuming, and we wanted to use another strategy to create SH3-TC2 models. So then we use the CRISPR-Cas9 strategy, and we obtained 2 models, one with the most frequent mutation at the homozygous stage 2, and another model with the mutation that is present in the spontaneous mice model. So we publish these results. So we've got iPS cells. Then we have to differentiate them into Schwann cells. And we did it for the control, but also the mutated cells. So we did the differentiation, you can see it. And after 30 days, we were able to have immature Schwann cells. So we wanted to check if there were the expression of mRNA SH3-TC2. And so here you can see the expression of SH3-TC2, but a relative expression compared to the expression, basal expression in U2OS cells. So what you can see in control and in the mutated cells, in iPS, there is no expression at all of SH3TC2. But during differentiation, you can see that SH3TC2 here in blue in control, you've got the expression started from day 24, and that at day 30, we've got a very nice expression. When we look at the mutated cells, you can see that we've got a reduced level of SH3TC2. Then we wanted to be sure that we've got Schwann cells. So we tested these genes that are supposed to be expressed in the Schwann cell. And in control, we've got a good expression. But in the mutated cells, we've got a reduced expression, like if there is a delay in maturation. So, okay, it was for mRNA. Now for protein, what do we have? I show you here the SH3TC2 protein and the two mutations that we are interested in, and the antibody we use to perform Western analysis. So here you can see that in control, we've got the SH3TC2 full size as expected, and we do not have, also as expected, the full size in the mutated cells. Then we use another antibody targeting this area, and we perform immunofluorescence. And in control, you can see in red the expression of SH3TC2. Then here you can see in the most frequent mutation model that we've got a reduced expression and maybe a mislocation. And in this model, the antibody is not supposed to detect, so we've got no detection at all. Okay, so you will say, okay, but SH3TC2 is involved in myelination. So currently you've got only Schwann cells alone. Okay, so we try to put together motor neuron and Schwann cells, and we wanted to obtain myelin. So we start with co-culture in 2D co-culture. And what we did is that we plate first Schwann cells in the plate, either control one, either the mutated one. And then we add control motor neuron on the plate. So here you can see in the confluences, of the motor neuron when you grow the motor neuron on the control Schwann cells. So we've got a very nice confluences that is seen in white. And in the mutated, when using the mutated Schwann cells, you can see that the confluency of the control motor neuron are reduced, and is significantly different for these models. Then we check at the hub size that you can see on the motor neuron, and you can see the size of the upside in control and in the mutated models, and we've got a reduced size of the hub, especially for this mutation. Okay, then 3D culture. So we did the first step exactly the same way, and then we tried to have it in a ultra low attachment, so that we've got spheroids, and we put them then in matrix gel. And what we obtained very nice myelin, as you can see, with a very compact myelin when we use control and control. When we use the mutated Schwann cells, you can see that we've got non-compact myelin. We see this one we've got a multiple observation for this one and less frequent than in control for this one. Also published. So my take-home messages are these one. There were no model, and now we've got human iPS harboring mutated strand cells that we can use and that we can share. We will be more than happy to do a collaboration. And as you can see, you saw, we are able to do 2D and 3D co-culture with these SH3-TC2 models. So we will be able to better understand the pathophysiology, but also to test drugs that we already started to do and that we would like to continue. So here is a team that I work with, and I would like to especially thank Camille Lauré, that was my PhD student and who performed most of the experiments I showed today. Thank you very much for your attention.